pushing us online. It's a bit slow this morning. Don't know why. Sorry, I forgot to charge my headphones, so there's a bit of background noise on my head. You may hear some French every now and then.
You're in Bordeaux.
I'm in Bordeaux, yeah.
Okay, so I sent the link to the bioarchive paper. Can you guys see it?
Yes, we can. Thank you very much.
So I'm kind of kept on time because our meeting here, which is in the same room, but starts at 10. So I think.
Right, no introduction and take it away.
If that works for you.
Yes.
Right. Then I will share my screen.
Ah, Katja's paper. Cool.
Yeah, so it's maybe a short introduction from my side. So I'm Sjoerd, I'm a postdoc at the NIN, and I've been eavesdropping on these journal clubs for a couple of months now, I think. And you guys asked me to present a paper, so I've seen you guys present a couple of papers, so I hope I'm doing it in the same style. Can you see my screen? So you see the paper? Cool. All right. I see some people nodding, so I take that as a yes. So the paper is called Principles of Neocortical Organization and Behavior in Primates, and it's indeed by Katja Hoyer and Roberto Toro. And since there's a whole bunch of French people in this group, I suppose you know the Institut Pasteur and a bit of where she's coming from. Basically what the paper is talking about is she published previously data sets of MRIs, post-mortem MRIs of different primate species. So not just usual suspects like rhesus monkeys or humans, let's say, we are also primates, but she did this for 70 different primate species. And then overall, she and Roberto Toro together, they have this theory called mechanical morphogenesis. So the fact that the folding of the brain is not just shaped by experience and by genetics, but that there are also some physical constraints which explain this kind of folding pattern that we see. So that's also, I'm going through the abstract now, so that's also kind of, you know, what they talk about. So they talk about the role of genetics, experience-dependent processes, which of course in developmental neuroscience for most animals are most important, right? The genetics and all these molecular tags and cellular mechanisms, they kind of shape the basic organization of extant of the brain in general. But then they argue that this view is incomplete, so that it's not just genetics and experience-dependent processes, but that there's also this theory that they have this mechanical cap, let's say, on top of that. And how do they study this, or how do they prove this? That is by doing what they mention here, independently of phylogenetic position is what they prove. So they want to look at different primate species coming from different kind of lines, and then see how their cortical expansion, how that's evolved separately. And then attached to that in this paper, they also look at behavioral correlates. So whether the folding and the size of the brain is related to the independent behaviors of these primates rather than phylogeny. And then one of the words here that they keep on, that I kind of like, is this capuchin anomaly, because they kind of focus on the capuchin monkey, which is a New World monkey in South America, with New World and Old World. A bit archaic, I would say, referring to where the primates come from, where they evolved. So we are kind of New World primates, let's say. And the capuchin anomaly kind of shows, that's also what they show in this paper, it's a monkey that evolved in South America mostly, but has similar, like, elaborate cortical folding patterns. It's very much comparable to macaque monkeys, for example, and much less like more genetically related monkeys in South America, like spider monkeys or marmosets. So yeah, that's the outline of the story. I think going through the introduction, I can, of course, but it mostly addresses what I've already been talking about. And the main thing is here, this New World and Old World monkey split that they study in this publication, or well, in this bioarchive thing. And this split happened around 47 million years ago. So she also has this beautiful video that I very much recommend everybody to watch. But I won't show it here, where she basically they basically show the brain size of the monkeys increasing independent of phylogenetic position. And you can see that it's very nicely morphs into more cortical folds, which is kind of independent of how they evolve. So let me go to the figure. I think that makes most sense to start with or to discuss. So that's this first figure. So that's, I think it's mostly very pretty, actually, but also informative. So what you see here are basically the 3D models of the expanded cortex, right, of humans, but also of other primates. And then they are grouped, color-coded by their different positions in the phylogenetic tree. So you can see the apes on top, right, so humans, but also bonobos, chimpanzees, gibbons. And then you can see a bunch of monkeys below that. So some old world monkeys like olive baboons, Japanese macaques, for example. And then all the way down, you see more New World monkeys like black spider monkeys, Colombian spider monkeys. So that's how they're basically grouped. So that's what she studies, right? She studies these, or they study this, these kind of cortical patterns and also cortical thickness. Of course, with these 3D models of these post-mortem scans, you can do a lot of things, of course. So yeah, that's basically what they show, right? And it's grouped from the largest on top to the smallest at the bottom of all these different primates. So I think that's a nice first illustration. But then this figure is more important. So here, basically, they show split, right? You're in the middle, this 47 million year ago, that the monkeys split from the New World monkeys, which was around the same time, which I thought was kind of cool to illustrate that hippos separated from whales, so quite a long time ago. And then, yeah, you see the phylogenetic tree here, and then what they mostly, yeah, but you can see what you can appreciate here on the bottom left in B is, of course, this split, and then you can kind of qualitatively see that the cortical patterns evolved in different ways, but in a quite similar way between New World monkeys and monkeys. And basically what they argue is that this tufted capuchin has a very similar cortical pattern, folding pattern to crab-eating macaques, for example, and much less like marmosets within the New World monkeys. And then in C, there are a bunch of other examples that are like this. So that, oh, sorry, I assisted, but I did not. To the contrary, brains of different sizes will have very different folding patterns, even among species from the same family, right? So that really depends more on the size than on the fact that driving the same point home, basically, than on the fact that they're from the same kind of family and the same genetic background. If there are any questions so far, then, oh, we do it at the end, right? Yeah, let's do it at the end. It's easier. So then they use this several different quantitative measures as well, right? So what they do here in this figure is they show the evolutionary history of brain phenotypes, right? So they plot the time at which they've been split, the x-axis. So 47 million years ago was the split between all the New World monkeys. And then also the absolute brain size. And you can see that it doesn't necessarily go one-on-one, right? It's not that all New World monkeys have smaller brains than all Old World monkeys, for example. And that there is a bit of crisscross within these genetic lines. And then on the right, there are a couple of more measures. So besides absolute brain volume, they also take brain surface and split it up. And this time, it's the time that they split is on the y-axis rather than on the x-axis. So you see surface area shows kind of similar pattern. The gyrification index, I'm not going through exactly what it means, but it has to do with the folding and also folding length. So these are different indications, quantitative indications, measures they take based on the structural MRI, and then they compare all these different species. And as you can see, for example, the capuchin, that's why they go into this capuchin anomaly in many of these indices is very close to some of the old world monkeys. So then here are some more quantitative, so more data analysis, let's say, on these patterns. So first of all, well, they look at similarity folding, thickness, and connectivity patterns across species. So in the top left is basically what we've already seen. You kind of see the 3D model of these different species. And in the middle, there are these flat maps. And then what they do in B, and then what they do in C is that they take these correlation values, and I have to reread a little bit what they did, but they have a correlation matrix that shows the similarity of the folding patterns across species. So then they kind of compare the two and see how much they relate. And then they see the same message basically, that they see that these if it's independent of phylogeny, you can see that the patterns of these monkeys with larger brains correlate better than the ones with smaller brains, marmots and owl monkeys in this case. And then they do a similar thing this time for in D. They use the similarity of cortical thickness patterns, which you can see kind of D is more an illustration, and then in E you see like it's a density map, right? How thick it is and how it's distributed throughout the brain. And then when they correlate these maps, these different brains with each other, they see kind of the same pattern as you see in the first row. And then the last analysis was a bit more complicated from what I remember, but they did like connectivity patterns. So they used, I think it was DTI, but they kind of randomized. These are not like regions of interest that you would normally see in a brain map, right? It's not like Brodmann areas or something, but they randomly parcellated it based on connectivity profiles. Then they reshuffled, and then they also correlated the different brain maps to kind of see how structurally similar they are, and then they find the same kind of pattern. And then so then the last thing, and this is important, this is not a figure I would like to skip because this is one of their important findings, is that they kind of took a bunch of behavioral indices from the literature, right? Because you can imagine these monkeys have been studied in primatology work, for example. I think it's easiest to start with G to appreciate a little bit what kind of factors people look at. So whether the animals are terrestrial, these primates, whether they use social learning, tool use, but also that's all the way in the top here in figure G, figure 5G, but also that, and these are things that were very predictive, but they also looked at average group size and all the way to the bottom to like what kind of diet these animals have. So they used a bunch of these correlates, these behavioral indices. And then what they argue in A, so they plot the neuroanatomical distance, so the difference in neuroanatomy with the behavior, and of course, one would predict that there's a clear relationship, which they find overall, right? But then what I understood from the analysis that they use is if they use phylogenetic distance as a covariate, it has way less of a predictive value than neuroanatomical distance. Again, saying that this, the folding and the neuroanatomy is more important than how related the animals are. I mean, of course, there's a relationship between phylogenetic distance and neuroanatomical distance as well, but you can take that out and then you'll see that it's not the most important. And that's what they do in C, if I'm not mistaken. Well, yeah, there's a relation between the two as well, right? Neuroanatomical distance and phylogenetic distance, what I've just mentioned. So these are like A, B, and C here to the left. And then in D, they kind of, as I understand it, they show like an overview of the total effects, how they relate, right? So then basically a summary of what I just mentioned. So behavior and phylogeny are not so closely tied as behavior and neuroanatomy, and phylogeny and neuroanatomy. And then in the middle in E, they take these behavioral clusters, right? And then they compare the more, then as you can already appreciate, right, just from the image, is that the brains and the cortical folding patterns look very different based on this behavior, right? You can see in the second cluster that there are way more animals with bigger brains, but also more folded highly folded brains. Um, yeah, and that's basically what you see in F and G as well, that there's a clear difference between these clusters, but also that there are some components, some correlates that are very predictive for this relationship, such as social learning and tool use, and some of them that do not seem to matter, whether the animal is nocturnal, polygynous, or something. So yeah, all of that combined to basically show kind of this main effect, right? So that it's that what they want to establish is this mechanical cap, this mechanical theory that puts a limit on how the cortex evolves, and that this has happened through convergent evolution. And then in the discussion, they go a little bit more into a couple of other options why this may have happened. Um, so maybe one of the things that they speculate about is that there were similar selective pressures that led to this cortical folding pattern, so that it was not the mechanical cap, but the natural selective. But they argue this is very unlikely because of the plasticity that occurs, and yeah, that it happened in exactly the same way is kind of unlikely. It seems that's what they claim here. And then let me think whether there are other points that I thought were worth going through. Um, yeah, what I liked is, of course, that they use the MRIs rather than the endocast. So they use endocast for a couple of reasons. So not sure how familiar people are with endocast, but it basically means like for fossils, right? You don't have fossilized brains because it's, of course, wet tissue. So then they take kind of the outline of the skull and either fill it up, literally fill it up, and determine the volume, or they use a CT and then estimate how much is in there. But of course, that isn't always as accurate, right? Dinosaur endocasts, they can have huge skulls, but actually their brains are kind of minimal. So are expected to be kind of minimal because we don't know, right? But if you look at Nile crocodiles, that's kind of the case. Then they go about this really cool talk about this really cool ancestor of all monkeys, Chilicebus carasunensis. It's kind of cool if you look at it online. I think what they estimate that the common ancestor looked like. Um, and then here another theoretical consideration that they also talk about is, there are people who believe, perhaps researchers that believe that there is kind of a potential in the genetics of some of the primates, right? So that some primates, the primates that are our ancestors already had the potential to have in their genes to develop further cortical folding and these kind of patterns. So it's kind of in a dormant state, but they consider this kind of unlikely when you think about the evolution of natural by means of natural selection. Um, yeah, I think they discuss a bunch more, but perhaps we can, discuss these last 10 minutes. And if there are any questions, I'm not sure if I can answer them, but at least we can figure it out together.
Thank you so much, and thanks for volunteering. Chris, take it away.
Hi, thank you for presenting this really cool paper. So two things. So they relate these changes, or they correlate the folding of the brain with some behavioral variables, right? But they don't look at subcortical structures. It's only the cortex, right? Yeah. So I feel that would also be interesting to see if folding could be also related to. 'Cause I kind of feel that if you had the biggest and most folded brain, but no subcortical structures, I'm not sure you would have the same like I'm not sure you would be immensely more intelligent, and I think because like some animals have much bigger brains, And I mean, we don't know. We can't discuss with them, and maybe they have the craziest philosophies and models of the world or whatever. But so far, it doesn't look like they have such complex behaviors as primates even. So I don't know if there is also a matter of the subcortical aspect, but I don't know if they discuss that.
I did not see that in the discussion. What I would think though is that in, but I could be wrong here, is that the subcortical structures are less constrained by this expansion factor, right? They are in the center of the brain, and the cortex is really where it gets, so kind of kept by the constraints of the skull.
But they're
So that's
also like
where they
connected
look at.
to like pretty much everywhere in the cortex. So
True.
they have a role with the cortex. So what if you don't have that hub? Like a lot of papers, especially with functional connectivity and stuff, they talk about the hubbiness of the subcortical structures. So would that be an advantage to grow a bigger brain without these subcortical areas? That I like. That's what I'm questioning, because like I don't know. I don't know if we have a lot of subcortical nodes compared to other species. I don't know if that's a factor, but that might be an interesting thing.
I agree. I think it's kind of a different paper, but
Yeah, that
yeah,
would be a
I
different
agree.
study. I just wanted
Yeah,
to
I'm not
like
sure.
see
I'm
if
not
they
sure about
discussed it or
the.
not.
Yeah, I haven't really seen it. Could have. Yeah, no, I haven't really seen them in the discussion. Yeah,
Thank you.
thanks.
I don't know who was next. I believe it was Fernando. Hands were
I thought
flying
it
too
was
fast
Hervé.
today.
My turn, okay. Um, yeah, thanks, Jerd, for the presentation. That was interesting. I wanted to ask your opinion on something. So just speculating.
Sure.
So if I understood correctly from the paper, if we have different, mm, yeah, different species, but having displaying similar behaviors, then we can also observe similar brain structure in some way. But I'm also wondering within the same species, so chimps, for example, um, would you say that if we compare wild ones with, like for example, in different environments of chimps that maybe require different sets of behaviors, would this then reflect that this chimps that are in these different environments will have more similar brain structures within themselves?
I think so, but this is also something they mention in the paper, right? It's first of all, it's genetics, which causes development of the brain, but it's also experience, mostly early in life, is what I would think. Um, but of course, that also shapes the way the what the brains look like. Whether you will be able to see differences in brain overall structures in between captive chimpanzees and wild chimpanzees, I don't think so. I think those would be very subtle, right? Just like, I don't know, human cultures growing up in different ways. That's probably gonna also have to do with stress. But no, I guess I don't think you would. It would be easy to see that. I think the difference between individual chimpanzee brains would then be probably bigger based on personality or other traits than these kind of experience factors. But to be honest, I don't know. I think, yeah, I don't think it would make such a big difference.
Thanks. Yeah, it was just
Yeah.
a little thought experiment.
Harry.
Yeah, my turn. So thank you. So a really interesting paper. So I need to read it. So I didn't read it yet, but I'm definitively do it. So it's a. I think there is questions about. So it's a comment and a question after. So I think the I like the idea that because as you said, the genetic is the point of starting point. And I think Eduardo wanted to know, there was a question in the chat. So, and I like the idea that the genetic is starting to change, and is the starting point as the cellular, the mechanism, the expression of the genetic expression on the cellular mechanism start, but the mechanical constraint on the and I really like this idea that the mechanical constraint on the folding of the cortex change the expression of the genes. So that means, yeah, the genetic playing a role, but also the environment and the genetic the folding. I think it's one of the idea of Roberto and Katja.
True.
And that is a comment I had. So I really like this idea that genetic is not doing everything. And so my question is, do they talk about, because there is a lot of studies looking at heritability of sulci and foldings. So I don't know if they talk about it and how they. I'm pretty sure that we can make a story about these two things, but there is studies showing that there is a clear genetic heritability of the sulci. So so I don't know if they touch this point.
Yeah, I'm not sure. You mean intergenerational, or do you mean from?
Even
that's of course not what they do. But...
Yeah, because here the genetic entry is the phylogenetic.
Yeah, exactly.
So, but in human, for example, if you do heritability studies, you will find that sulci are really heritable. So I don't know if they talk about.
It's a good question. I don't remember. I mean, they definitely do not have the data, right, to do anything like this.
Yeah, for the interspecies, yeah, it would be difficult to do it. But
yeah, definitely, and it's also not really there. But I don't remember them talking about that. They talk a little bit about how plastic the brains can be, right? So you change a couple of genes and it can change dramatically.
Yeah.
In terms of what the brains look like, and it's also developmental disorders, of course. I don't remember them talking about it in this context.
Okay.
Specifically.
Because I think the next step will be the interplays in the single species. It will be the interplay between this mechanism, the anatomical or mechanical mechanism, and the genetic mechanisms.
Yeah, I agree, and potentially also how this mechanical, these forces shape genetic expression, right? That's also definitely possible in.
It's also, a circle.
They argue. That it relates. Yeah.
Thank you. Very cool.
Yeah, I thought it was exciting. Maybe a bit different from other papers.
Definitely, and I can see people are coming into the room, so we're gonna let you go.
Yes, we are. I'm onto my next meeting, but it was fun to present. Thank you for that.
Thanks for volunteering, and we have Irene present 2 weeks in a row, next week and the week after. Thank you for that. Have an exciting week, everyone in science, and see you next Monday.